have to switch what I'm thinking about, but I'm always very, very excited about talking about research and plants in general to people who are interested. And I'd mentioned one of the things that I do is I teach a class in science writing. And I think in general, we scientists usually do a terrible job of talking enough about what we do and getting enough people excited about it. So thank you for giving me the opportunity to do that. I'm obviously not quite sure what the background is of people. I know that you're all excited about nature and very curious, and I think that's pretty much all you need. But if I talk about things that don't make any sense or you have questions throughout my presentation, then please speak up and ask. I will try to keep it short, which is not always easy, so that there's enough time at the end for some questions. So my background is in plant ecophysiology, and I spent some time in Panama, in the rainforest doing research. I have worked in California before coming to Wellesley, and I've always been interested in how plants manage to deal with the environment, because they are sessile, they're sitting there, temperatures are changing, light intensities are changing, and they sort of have to make a living that way. They cannot just run away and hide somewhere. And I worked on a variety of different things. And when I was in California doing a postdoc, I came across a picture of these leaves with, a face of a human on it. And that sort of got me interested in what this is about. And it turns out that what you're seeing here is actually a leaf, a real leaf. This is not, manipulated on Photoshop or something like that. But this is really a leaf that has patterns where there are darker areas and lighter areas. And the reason for that has to do with the fact that people took a leaf, took a transparency. I don't know if you remember what that is, a clear plastic foil on which a picture in black and white was printed, and they put it on top of the leaf and were shining light on that leaf. And in response to that, the chloroplasts were moving, the chloroplasts being the organelles in which photosynthesis takes place. And there are many of those in each cell, and they were moving in different positions, and that influences what the leaf looks like. So you actually can see chloroplast movement live in the real thing if you do something like that. Doesn't work with every species, but this may be a fun thing to try out in your group at some point. But that is basically how I got interested in chloroplast movement behavior, because I saw a picture that I thought this must be fake and whatnot, and started reading about it. So I want to tell you a little bit about what actually chloroplast movement is. And I have a picture here taken by some people in Germany at a university, where they are looking at an algae, so very thin leaf-like structures. And you can maybe make out their individual cells here, and then the green things, those are the chloroplasts. And this is the same algae, the same part of the algae under the microscope. If you're shining very low light onto the algae, what you will see is that the chloroplasts start spreading out, right? They're neatly spaced out to maximize how much light they can absorb. And people call this an accumulation response. They accumulate in low light. They put their big fat side up, and they try not to overlap so that they can get as much light as possible. If, however, you're shining strong light on this part of the algae, then it looks totally different. You still can see the green, the chloroplasts here, but what they have done is they have moved to the edge, and they have been diving down. They pile up on top of each other, and they are avoiding the light. And this is called an avoidance response. Now, people have known this actually since 1880 or something roughly like that. In fact, there was a book this thick published by Gustav Sen in 1908 that was filled with all sorts of knowledge about chloroplast movement, drawings and showing it in different species. So people have known about this for quite a long time. Like everything in science, you know, things go in phases. So it was in and then it was out. And then in the 50s, 60s, a lot of people picked it up again, mainly in Germany, a lot of people in Japan. And they figured out a few things like that the chloroplasts actually respond to the blue light, that wavelength range of the light, not to red light or green light or something like that. And there were some advances made. And then again, it became sort of not, didn't make more progress and it stopped. 25 years ago, some people started getting back into the literature and saying, hey, this is a cool topic, but now we have different tools and we can do so much more, you know, let's get back and sort of see, you know, can we answer some more questions about what chloroplasts and their movement. And so some of the questions that are obvious, right, is how do the chloroplasts know when to move? Okay, we know it's something to do with blue light, but still, you know, what is it? They don't have eyes the way we have. How can they sense light? And then you have that signal, the light is the signal and says, okay, it's too much light or too little light, but you know, then you need a pathway, that message has to be relayed in some way. The chloroplasts have to be told, okay, now move, right? And then the other question is, how do they move, right? I mean, a chloroplast is in the inside of a cell. You know, is it pulled or pushed, or does it crawl, or you know, what exactly is it that it does? And to answer a lot of these questions, you know, one would probably look at the genetics behind it. So I will tell you a little bit about that. And then with many behaviors, you know, the question is always, is the behavior stereotyped or context dependent? Meaning, if I take light and I shine it on a plant, will it always behave the same? Or is it context dependent? Does it depend on what species or how it grew up or what happened to it earlier in the morning in that day? And is it universal, right? And then in the end, you know, if you're a plant and you have chloroplasts and they're moving around, you know, I mean, is that actually helpful? I mean, it seems like a lot of energy is expanded on that. You know, does it help with dealing with life and growing and fitness, which means how many offspring you can have, right? And so I want to touch a little bit on all of these topics. Some of them are not things that I did. Part of it is stuff that I worked on with my group. But before I can do that, I want to at least give you a little bit of a flavor of how people can approach questions like that, because it's maybe not as intuitive as, you know, going out and counting number of monarchs, or, you know, getting your binoculars out and watching birds behave, right? Like, how do you know actually that chloroplasts are moving? And so the way you can do it, one way is microscopy, right? So I have a picture here of Arabidopsis thaliana, a cress that is a species that plant biologists love to study because we know a lot about it. We know its genetics, we know its genes, you know, a lot of things like that. And you can take a leaf here and you can shine blue light on it, right? And then you make a cross section, you put it under the microscope, and you look at it. And this is what these two researchers actually from Poland did, and they were the ones who got that whole field started again. And what you can see, this is a cross section to a leaf, right? So if I have my leaf like that and I cut it this way, lop it down, look at it under the microscope. So I'm looking at the top of the leaf, the bottom, and then these are the individual cells in it. The dark things, those are the chloroplasts, right? So if I'm shining low light on it, blue light, I can see the chloroplasts spreading out, accumulating. There's some at the bottom, but many of them will be at the top of the cell. So the light is shining here, right? And they're trying to be in each cell to be as close to the light as they can. If
Martina,
I shine,
I have a really quick question from someone. Can you just quickly remind us what a chloroplast is?
So a chloroplast is an organelle within a cell in which photosynthesis takes place. So you all know that plants can take CO2 and light and turn it into sugar, right? And the chloroplasts are the parts of the cells, the organelles in which this happens. So the chloroplast is what give a leaf its green color, right? And they are the machinery, basically, or within them is the machinery to do photosynthesis.
Fantastic.
Right? So,
thank you.
So by them moving, the plant can sort of influence how much light it is getting, right? So if it's in this position here, what that means, the chloroplasts are spreading out, they can absorb a lot of light. And if they absorb a lot of light, then that means they can do a lot of photosynthesis. They make a lot of sugars, they can grow and do all of that, right? But what's important is that there is such a thing as too much light. And so if you're shining very high light on it, what will happen is that the chloroplasts hide on the side of the cell. So again, this is, they're green, right? But we're seeing it in this picture as black, these roundish parts here. So they now go to the edge of the cell, they pile up on top of each other and shade each other, right? So we know that we humans do this, we go into the shade, into the sun, depending on what we want, but this is happening within a plant, within a leaf, within a cell, that there are all these little organelles and they are moving around to optimize how much light they are getting, yeah? So these are my chloroplasts here. And again, this would be an accumulation response, and this is an avoidance. So accumulation means get as much light as you can. Avoidance is you're getting out of the sun, right? You can also do this. So you can do this if any of you guys have a light microscope available. You actually can look at the chloroplasts in some species under the microscope, and you can see that. If you're lucky and you're at a rich place like I am at Wellesley College, you maybe even have a fancy microscope. There's a microscope that's called confocal laser microscope, where you can sort of get like three-dimensional pictures of leaves. And in this case, these parts, the light colored parts, those are my chloroplasts, where photosynthesis takes place, and you can study where they are, where they go, and things like that. So if you're studying chloroplast movement behavior, you will spend a lot of time in front of a microscope, right? And you can even make some movies. So I wanted to show you this movie here. In this one, our chloroplasts are red. So all of these colors are false colors, and they are red. So this here is one cell, right? And you can see that in one cell, there are many, many chloroplasts, and they're somewhere. So what I will show you is sort of snapshots of what happens when chloroplasts move. Okay, so I have, if you maybe look at this cell, you can see there are chloroplasts here and they are spread out. They are accumulating because there's not that much light here, right? If I put light on, let light shine on it, you probably missed this if you blinked, right? You can see the chloroplasts moved, right? So I'm going to do this slowly. You can see if you follow maybe one chloroplast, they were in the middle and now they're going towards the edge, right? Some of them are faster, some of them are slower, and then eventually they will actually dive down to the edge, right? So you
question.
Yeah.
Do the cells change shape as well?
The cells do not change shape. So remember, plants have a cell wall, right? So that is fairly solid. If it looks like they're changing shape, it has to do with the type of microscopy that we're doing. You're actually only seeing the chloroplasts and not the cell wall. I only know that this is a cell because the chloroplasts are usually at the edge of a cell. So that's actually a great question. It looks like it, right? Because when you're used to normal microscopy, you see the outline of the cell. In this type of microscopy, you only are seeing the chloroplasts. Okay, so doing this kind of microscopy is really fun. It's cool. It's visually appealing, you know, it's really nice, but it's the confocal laser microscopy where I showed you the video and stuff. That is a very time consuming. It's very expensive piece of equipment, so we don't get to use it that much. It's a shared piece of equipment. So we wanted to have something where you can investigate this much more easily and you don't need expensive equipment for it, right? And so just to remind you, if I have a cell, right, and I'm shining low blue light, very weak light, right, not much on it, my chloroplasts, in this case in green, will spread out to get as much light as they can, right? If I shine high blue light on it, they will pile up on top of each other at the side of the cell, and the cell shape won't change, right? And this is sort of the reason behind why, if you look at an image like this of a leaf, you actually can really see differences between lighter and darker areas, right? So the light area, like of the hat of the person here, this is an area where there was a lot of light shining on it and the chloroplasts are hiding on the edge of the cell, right? So what I can do is I can try to quantify that. And one can do it by shining red light on it. And then you measure how much of that light appears on the other side of my leaf, right? So if I have a leaf and I have thousands of cells with lots and lots of chloroplasts in them, and if all of them are in this accumulation response, right, and I'm shining light on it, then very little red light will come out on the other side, right? But if they're in this position here, what will happen now is a lot of that red light will go through to the other side, okay? So this is called transmission. When light hits a leaf, some of it is absorbed and used for photosynthesis. Some of it is reflected, that's why we see it, and that's why it looks green. And some of it is transmitted, meaning it gets all the way through the leaf, right, and is coming out on the other end. And so you can exploit that to actually to measure and figure out if chloroplasts are moving or not, right? The bad thing is that you cannot go to a store and buy an instrument that does it, right? Chloroplast movement is not like doing cancer research. There are not that many people that are interested in it, and most people use a microscope. And so we wanted to do something different. And I just wanted to show you this. You know, I'm an ecologist, right? Field work and stuff like that. But I was so fascinated by chloroplast movement that I wanted a way of measuring it, because as a scientist, you always want to put some numbers on things, right? I mean, you want to know what it is, how many are moving, are they moving fast, slow, whatever is going on. So we had to build our own instrument. So I got to work with a physics professor. I had some students who are engineering majors, and we built this instrument that allowed us to measure chloroplast movement. So we still do microscopy, but we also use this, and you can see lots of cables and chips and 3D printing and whatnot, a whole learning experience for somebody like me, but a lot of fun. Okay. And so what you can do in the end is you can measure transmission as an indicator of chloroplast movement, right? So what you see here is I took a leaf, right? And I was having it in the dark. So down here below is sort of what I'm doing to the leaf, what kind of treatment. So I have the leaf in the dark and I can measure transmission. So I find that 12% of the light is neither absorbed nor reflected, but makes it right through the leaf, right? If I shine weak blue light on it, the transmission goes down. And what that means is my chloroplasts are spreading out so that they can absorb more of that light. If I shine bright light on it, they change their behavior. They go to the edge of the cell, they're avoiding, and now more light is transmitted through, right? And if I increase the light even more, then more chloroplasts wake up and they go away and move out of the light, right? So I'll only show this once to you, but I wanted to give you a feeling that if you're studying something that is actually happening in the inside, obviously, of a leaf, right, you have to be creative. You have to come up with ways in which you can investigate it and sort of figure out, you know, what is going on. And like any good scientist, you can be quantitative and calculate ratios and slopes and, you know, whatever things like that. So once you have a method like that, then you actually can try and figure out, well, where are the eyes in the plant, right? How does it know blue light is coming, or that it is high blue light, or things like that, right? And there was a group from Japan who were some of the first ones who used this method and found out some details about this process. So this made it to the cover of Science, one of the most prestigious journals that are out there. And this symbol is the Japanese symbol for light. And so they projected it onto the leaf. And you can see that in the areas where it's light, the chloroplasts move to the edges in an avoidance response, right? But they used an instrument similar to the one that we developed to get some hardcore numbers and things like that as well. And if you do that, then you can look for mutants. So a mutant is an organism that is messed up in some way. You're changing its genetic makeup, and you're doing it in hopes of finding out which molecule is responsible for what. So I'll just show you this here. So any normal plant, Arabidopsis plant, if you're shining low light on it, it will spread out its chloroplasts. You probably know by now, right? If you shine bright light on it, so the big arrows mean bright light, then they hide at the edge, right? But the scientists found some mutants where they could, this plant here could get out of the light, but it didn't accumulate, right? The chloroplasts were not lining up right? Or in this plant, they lined up right under low light, but not under high light. Right? And this is how scientists figure out the molecules that are involved in a process. So what they found is actually the light detectors, right? So we have light detectors in our eyes, but our eyes are in one specific part of the body like in most animals, right? In the plants, these little sensors are in every single cell. Okay, so it's decentralized. They're not in one specific part, but every part of the plant can sense blue light and send a signal to the chloroplasts and make them move. Okay. Another question is, okay, the chloroplasts get the signal and they somehow know, okay, there is low blue light, which means, I probably have trouble doing photosynthesis at the rate I should be doing it. Let me go and look for more light. Let me move towards the light, right? Or they're getting a signal, oh, strong light, I need to get out of here. If I stay here, I will be damaged and this will be really bad for me, right? And so they're moving away from the light, right? So people actually have figured out some of those signals, what is the language that tells the chloroplasts what they should be doing? Should they move towards the light or away from it? And I can talk more about that if people have questions. But the next question is, how do they move, right? If you think about animals and movement, you're probably thinking muscles, right? So if I'm moving, if I do something like that, my muscles in my legs, if I'm walking upright, right, they are contracting or relaxing. That's how animals move an entire body, right? Well, plants don't really move. They can grow towards light or away from it. But in the inside of a cell, you have to imagine it's really, really cool. There's like roadways. They're like tracks that run throughout the cell. And on those, so they are big highways that branch off into smaller ones, and along those ways, cargo can be transported. So if a plant needs to bring certain materials from one place to another, it can package them and can make them move along these roadways. Animal cells do something similar, so very, very cool. So scientists thought, maybe the chloroplasts use those highways, right, to move from place A to B. And looked for it and looked for it. I was one of those scientists who wasted, in the end, about 2 years of my life trying to figure this out. And it turns out chloroplasts are doing things very differently. It took, again, a breakthrough in technology for people to figure this out. A very cool group of scientists in Japan and Korea who have this amazing, amazing microscope. And what they could do is they could visualize at the same time the chloroplasts, and you're seeing them in red here, and then actin, which is a molecule that is in your muscles. So you maybe have heard that your muscles are made out of fibers, out of muscle fibers, and the molecule that makes them up is called actin. And what they found, they could make actin visible. It's colorless, you cannot see it, but they could make plants that had green actin in them, and they could look at it under the microscope. And what you maybe see is a chloroplast in red, right? And then green all around it. So the chloroplasts have their own muscle fibers, so to speak, all around them, okay? And they also can take videos, and they did that. And what they found is if I have a cell here where I'm shining blue light on this and I have my chloroplasts nicely spread out, can you see where this light was shining on? Now there are no chloroplasts. They have all moved to the edge, right? They sensed it and they moved away. And if you want to see cool videos, whatever, they have done some amazing stuff. So what they basically showed is that chloroplasts can move on their own. They have their own muscle-like structures, and they can go anywhere. They are not restricted to the highway system that exists in a plant cell, but they can move from here to here, whatnot. And this is why when you watch them, it's not like they going like this, right? But they can go up and down and whatnot in any direction because they have their own independent system. Okay, so and they can be pretty fast. I mean, you may think this is not that much, but a chloroplast can go at 1 or 2 micrometers per minute, but it's pretty impressive. Okay, so I just wanted to give you a little bit of a flavor that there's a lot of cell bio, molecular bio kind of stuff behind it, but then now switch a little bit more to the ecology part of it, right? So I worked on part of this, and as I said, we found some cool stuff, but also, you know, had a lot of, wasted in the end a lot of our time because we couldn't figure things out, right? And so we decided to go back and sort of, now that people know that chloroplasts respond to blue light, we know how to measure it, we know that they can move on their own with their own system, muscle system, basically. We wanted to see, does every plant out there do this, right? Is this something that is universal? And are all of the plants doing it to the same degree, right? Are there some that are chloroplast movement champions, right? They move their chloroplasts around much more or faster, or, you know, is there any patterns to it? So what we did is we went out just in front of the Science Center and collected some plants. Maybe you recognize the dandelion. We had water hyacinth in the greenhouse. We had different species in the greenhouse. So we looked at like 10 different species. We measured them. We looked at them under the microscope. We looked at transmission to sort of see, are they moving? We quantified it. We stressed them to sort of see, does moving chloroplasts help with stress? So the idea is that plants have to have a certain amount of light. If you have too little, you're not doing well. But if you have too much, it's also dangerous. So can they do things so that they stay unstressed, right? So we did a lot of different kinds of measurements. And again, sort of one big important thing is that probably many of you know, but it's not, if you, the more light, the better. There's sort of an optimum range. So if you look at photosynthesis, too little light, the rates will be low, and then at some point you're reaching your maximum. But if you're giving your plant too much, it actually will be suffering, right? And what we found was that different species behave very differently. Some species move their chloroplasts a lot, others don't, some not at all, right? What we also found out is the avoidance reaction is always faster than the accumulation response. And if you think about it, that I think makes intuitive sense, right? If you have too much light, like if you are sitting in bright sunlight, right? It's more important for you to get out of that so that you don't get, and quickly, so that you don't get a sunburn and some damage, right? If you're in a shaded area, you might, as a plant, do better with more light, and moving your chloroplasts so that you can capture more is a good thing, but it's maybe not as life and death kind of thing as getting out of the sun, right? And not surprisingly, what we found is many weedy species that are opportunistic, that you find in disturbed areas, they seem to be very good at moving their chloroplasts. But we had a hard time figuring out exactly sort of what was going on there. And it's probably because we looked at 11 different species, right? You know that there are at least 400,000 plant species out there, and 11 randomly selected species is probably, you know, not going to answer all of our big questions, right? So, because if you have different species, some of them were aquatic that we had, some of them grow in the sun, some of them were trees, some of them were shrubs. So I think we had too many different things going on at the same time. right? And so what we did is we actually went back and we used some mutants. The mutants, again, are these plants. They're all Arabidopsis plants. They all look the same. They grow the same. Everything is the same. But their behavior in terms of can they move their chloroplasts or not, that is different, right? So using that, what we could do is answer the age-old question of nature versus nurture. I'm sure you're very aware of that in the context of health issues, right? There's always the question that a heart attack, right? How much of that is genetics? Is it because I inherited some genes from my parents, and because of that I have high cholesterol, but it's all in the genes? Or is it because of behavior? Is it because of how I grew up? Because of what I do, what I eat, exercise, or things like that, right? And scientists are very much interested in that about everything. So what we wanted to know is, are there genetic differences between plants, that some of them can move their chloroplasts a lot or not? And or is it the environment? Is it important how you grow up? Because we saw that not every species was behaving the same, right? And so we set something up where we had Arabidopsis, our model plant, this little plant. You can grow lots of them outside in the growth chamber and all sorts of stuff. And we had wild type, meaning a normal functioning one like you would find out in the wild, right? And we had mutants, some of them that could not move their chloroplasts out of the light or into the light or both of it, and all sorts of different things. And with that, we could mimic what we would find in a range of species in nature, right? But we are doing much better now because they're all Arabidopsis plants. Everything else is the same. And then we grew them under different light conditions. So we're basically saying, well, let's have how they grow up and what they're used to. Let's change that and see if nurture, their experience as a plant, if that makes any difference. And then we looked at chloroplast movement behavior. We did stress and growth and all sorts of different things. And what we found out that it's both the genetics that's important and how they grow up. So, and plants that couldn't move their chloroplasts out of the light, they were much more stressed than those that could. And moving your chloroplasts out of the light was really important for plants in the shade. So shade and sun plants are those that have grown up in the shade, and then there's sun plants. If you're a gardener, you know what I mean, right? There's some plants that you plant under your trees and they really love it there, right? Others want to be out in the open and get as much light as possible. So surprisingly, the ones that are in the shade, for them, being able to move their chloroplasts around, that was really, really important. That wasn't really what we had expected. And then the ultimate question was, does it make a difference to how well they grow? And so this is what Arabidopsis looks like as a youngster. It just has some leaves that are in a circle, in a rosette. And at some point it puts out inflorescences, so a stalk on which there are a lot of flowers. And then it makes fruits that are called calyx. And so we had plants that were different in terms of their genotype, meaning they were genetically different. So everything was the same except their ability to deal with light and to be able to move their chloroplasts. And then we measured as much as we could. We grew them for weeks and we measured rosette diameter, meaning how big was my plant? How many leaves did it have? We harvested it. How much dry mass did it have? How many seeds did it make? How many fruits did it make? All sorts of different things. And we did this for plants that were grown in part outdoors in New England, hot, dry, cool, rain, drought, whatnot, and under controlled conditions where we changed the light, but we made it chilly. So this one treatment was like a summer experiment, the other one like fall, where it wasn't cold, but chilly enough. And we measured that. And what we found in the end was that if you cannot move your chloroplasts, then the plants don't grow as well. Okay, which is what we were hoping to find, but nobody had actually been able to prove that. People had speculated about that, but couldn't really show that. And what was also interesting is that there was an interplay between their genetic makeup and their environment. So overall, what we know now is that the chloroplasts, these organelles that are so important for photosynthesis, they move in response to blue light, so that they can, if there's too little light, they move into the light. If there is too much, they move out. They do this not using roadways that are in the cell, but they have their own motility system. So they have their own little fibers that can propel them to different parts within the cell. Not every species behaves the same. Some of them use this mechanism a lot, others don't. It's really important for the plants that grow in the shade. So if you want to actually see chloroplast movement, if you want to play around with that, take a leaf of a plant from a shade plant and lock out a little area on it and shine bright light on it. And then you should hopefully, after 10 minutes or so, see a pattern in the leaf in terms of the shade of green, right? And for this behavior, like for most behaviors, I would say, the genetic makeup is important, but also the environment, right? So you can have a messed up gene or something, and then you're behaving differently, but it depends on how you grew up. Did you grow up with cool temperatures or warm temperatures? Is it in the summer or in the fall? All of these things will make a difference. But if you can move your chloroplasts, you will be more stress tolerant, meaning you can deal with changes in light intensity more readily. You can grow better. And in the end, you can have more offspring. So that means this is an expensive thing to do, but it's definitely worthwhile doing it in the end. So those are my take-home messages, but I also wanted you to get a little bit of a flavor of how people go about doing things, and that very often when you do science, you really have no idea what you will have to learn or what you have to do. Like in my case, learning electronics and building an instrument, I had to learn confocal microscopy, I had to learn some genetics, all of that coming from an ecology side, because that was the only way to understand this issue really completely. And that's the end of my story, and I'm going to stop sharing, and I want to thank you for being